Subject I chose to cover for this video is secondary active transport. Now, active transport is the movement of substances either up a concentration gradient, that is going from an area of low concentration to an area of high concentration, or against an electrochemical potential, which is something like moving a negatively charged electron towards more negatively charged electrons instead of away from them. Because the movement of particles in what is essentially the wrong direction is an energetically unfavorable process, active transport always requires the input of energy, which is part of what differentiates it from simple and facilitated diffusion, which do not. Now, there are two types of active transport. There's primary, which is usually driven directly by ATP hydrolysis, and secondary, which is driven by a cell's concentration gradient, which I talked about earlier. Secondary active transport is also called indirect active transport. So although it requires the input of energy like primary active transport does, it's not as straightforward as primary or direct active transport is. In secondary active transport, the cell essentially makes a compromise. It moves two solutes or ions, which from here on out I'm just going to say particles because that's easier, simultaneously through a transport protein. But while it moves one of them up its gradient, which is what we expect from active transport, it moves the other one down its gradient, which is an energetically favorable process like facilitated diffusion. The combination of an energetically favorable process with an energetically unfavorable one allows the reaction to proceed with an overall decrease in free energy, a negative delta G, which allows for a spontaneous reaction. Now, there are two ways that this simultaneous transport can proceed. Either both particles can move in the same direction, which is called symport, or they can move in opposite directions, which is called antiport. Now, in symport transport, just as I mentioned, both particles being transported move in the same direction. Our textbook provides a good example of symport transport. In animals, sodium ions are at a higher concentration on the outside of the cell than they are on the inside of it. Organic molecules, on the other hand, and in this case I chose to use amino acids, are at a higher concentration inside the cell, which means that active transport is required to bring them into the cell. So to get the amino acids into the cell, they're coupled up with the sodium ions. Now, the graph from the textbook, which I was unfortunately unable to copy despite my best efforts, shows the correlation between the concentration of sodium ions on the outside of the cell and the transport of organic ion molecules into the cell. And it clearly shows how, as the sodium concentration increases, the rate of organic molecule transport also increases. Antiport transportation, on the other hand, like the name implies, is the simultaneous movement of two particles in opposite directions, although, like with symport, one is moving down its concentration gradient and one is moving up its concentration gradient. And for this example, I've chosen the exchange of sodium and calcium ions in cardiac muscle. And in this case, as you can see in my drawing, both ions are at higher concentrations outside the cell than they are inside the cell. So the movement of the sodium ion into the cell is energetically favorable because it's going from the area of high concentration to the area of lower concentration, whereas the movement of the calcium ion to the outside of the cell is going from the lower concentration to the higher concentration, but it's going in the opposite direction of the sodium ion. And this process is actually very important because if the calcium ions are not removed from the cells, your heart muscles would not be able to stop contracting, which would be fatal. So, while secondary active transport doesn't directly utilize ATP hydrolysis like primary active transport does, it's still ultimately driven by ATP hydrolysis because the sodium ions are pumped out of the cell via the NAK pump, which is a primary active transport. And that is what gives us the gradients that we need for secondary active transport to take place. So, without primary active transport, secondary active transport would not be possible.